Okay, so this is the stuff we're going to cover. I'm going to erase this because we need board space. Okay, so the first thing that I want to cover is the central dogma. The central dogma, okay, first of all, why is the central dogma important? It's important because it is the way DNA makes a protein. The way DNA makes a protein, that's what the central dogma covers. So the central dogma is something that's called the molecular central dogma of biology. It actually is DNA makes RNA, and that makes a protein. Okay, so let's talk about DNA a little bit. For students who have not, okay, everyone's heard of DNA. It's all on different, you watch movies and they say, oh, we looked at the suspect's DNA. DNA is everywhere. But let's talk a little bit about where this DNA is housed. We have a person. That person is made up of cells. Every cell in your body is made up, it has DNA in it, with the exception of mature red blood cells. So, these cells, let's take a biopsy, a piece of tissue, and this tissue is made up of billions of cells. These cells are like Legos. Let's do a zoom up on a cell. In a cell, you have a bunch of organelles. You have, of course, ribosomes, and you have Golgi apparatus, you have the ER, you have mitochondria. But you also have this really huge structure, and that huge structure is called a nucleus. The nucleus houses the DNA. The DNA, let's make the DNA look like this stringy stuff. The DNA is a blueprint. I've heard many books call it this. It's a blueprint of who you are. And it's a very important blueprint. As a matter of fact, the DNA is so precious that it doesn't leave the nucleus. It's like one of those libraries that I remember when I was going to UC Berkeley. There was this library that housed old books, and those books were so precious that the librarians didn't allow these books to leave the library. You had to go in, request a book, and they'd photocopy a portion of the book. They'd wear little white gloves. These books were precious. The DNA is like that. It's so precious. It doesn't leave the nucleus. But you can photocopy a piece of the DNA, and we call that photocopy of the DNA RNA. So the DNA, it's like rare books. They never leave the nucleus, the library. The RNA, it's kind of like a photocopy. Think of it like a photocopy of that rare book. And then with that, you can make, with the RNA, with this photocopy, you can make a protein. Okay, So why is this important? Why am I telling you this? It's important because we're going to talk about viral replication. And in any, um, so when a virus, we're going to talk about it replicating the entire virus. But when it does that, it has to make loads of copies of DNA for the new viruses. And it also has to go through the central dogma. It has to make DNA, and then RNA, and then the protein. The protein that the virus we're going to talk about, SARS-CoV-2, it makes a lot of proteins like spike proteins that coat the outside of that virus. Okay, we will go back to the central dogma. The next thing I want to talk about is actually the viral replication. Okay, so Dr. Nathan Peabody talked about this, you guys, but we're going to go over it very, very superficially. So we have the virus, and the one we're going to talk about is SARS-CoV-2, the one that, this is the virus. This actually stands for severe acute respiratory syndrome. Coronavirus 2, as opposed to the first coronavirus that the world saw in 2002. Okay. So this virus that causes the disease, COVID-19, this virus has an envelope that it actually stole from a prior host. It has spike proteins, studded with spike proteins. They're embedded. And it has, sorry for the noise, it has positive sense RNA. So we call this plus sense RNA. I hope everyone can see that. All right, darker. Plus sense RNA. So plus sense RNA is, it works the same as messenger RNA. We call that mRNA. This is RNA that doesn't have to be processed. It can just, once it goes into the cell, ribosomes will clamp onto it and you can start translating proteins right away. If it were mimesense, then the mimesense would have to be turned into plus sense, but this plus sense is ready to go. Okay, so that RNA, so here I'm going to draw a lung cell. The spike proteins actually latch onto a receptor. This receptor is called an ACE receptor. There's a proteolytic cleavage, and this RNA comes in to the host. So this host cell is the host cell. Let's make it a lung cell. So the host cell is a lung cell, and this cell will be forced to make what I call like baby viruses, and it hijacks the host cell, and then many viral particles are made, and this cell in that process is destroyed. So eventually we're gonna get a bunch of these viral particles that come out of this cell. So you see that this virus actually kills the lung cells. Okay, I want to move on to the immune system. So I'm going to stop here and we'll move on.